Mohamed Bentires Alj. I'm a professor at the University of Basel. The goal of our research is to understand breast biology and breast cancer and to use this knowledge to improve therapy for our patients in Basel and throughout the world. My lab focuses on the cell autonomous and non-cell autonomous mechanisms regulating normal and neoplastic breast stem cells, resistance to therapy, and metastasis. In collaboration with clinicians from the University Hospital of Basel, we are developing a breast cancer personalized medicine program. I'm pleased to welcome you to conversations with key figures in mammary gland biology and breast cancer.
Hi Mo, very nice to see you again. I'm Christine Watson. I'm working at the University of Cambridge in the pathology department, where we are now. And I'm a mammary gland biologist, and I'm interested in all kinds of things to do with mammary gland, but particularly cell death and lineage commitment of stem cells, particularly those that arise during pregnancy.
So Christine, we first met at the Mammary Gland Biology Gordon Conference in Rhode Island, I think in 2003, when you were vice chair, if I recall well.
That's right, yes.
And then I immediately felt welcomed by this scientific community. Can you tell us more about the spirit of this meeting? I think you chaired the meeting in 2004 in Italy.
That's right, yes. I mean, it was a wonderful meeting, I say so myself. But I think the thing I love about the Gordon Conference is it's so friendly that everyone is welcome. So new people coming into the field, we just love to welcome new people. And it's a great atmosphere. Everyone is very supportive. I've made so many friends at these meetings. People that I've known now for, 10, 12 years. And we've kept up our friendship. We see each other every year because we go to the conference every year. Absolutely. And I know you're going to organise it this year, which is very exciting because there is so much going on in this field.
Yes.
And it's a great opportunity to invite people from all areas of mammary gland biology and breast cancer.
It's one of the rare meetings that combines both normal development and cancer.
That's right. And I think, for my lab, every year I want as many people as possible to go because I think it's a fantastic way to get to know the field. So this year I'm hoping you'll allow a few people from my lab to come. But normally when it's in Italy, we try and send about half of the lab to the meeting because they all come back so enthusiastic and very happy because they've made new friends and learned a lot. And in the field, the field is moving forward so quickly. That I think you have to go every year to keep up with things.
Absolutely.
But it's definitely one of the must-go-to meetings every year because you learn so much and it's just such a lovely, friendly, stimulating environment. So good luck.
Thank you. Thank you.
I'm sure it'll go really well for you.
Thank you. So, Christine, you obtained your bachelor in biochemistry at the University of Glasgow.
That's
in
right.
1975. Yes. And your PhD in molecular genetics at Imperial College at the University of London. What was your PhD thesis about?
Well, my PhD thesis, actually, it's an area that's kind of disappeared because I worked on embryocarcinoma cells. And these are the sort of precursor to embryonic stem cells. So during the last year of my PhD, Martin Evans and others developed embryonic stem cells. So I was working on the kind of forerunner of that. And what I did, I was interested in looking at genes that were differentially expressed between undifferentiated embryonic stem cells or embryocarcinoma cells and those that were differentiated. And so I made cDNA libraries. And I was the first person in the UK to make a lambda-phage cDNA library. So now you know how old I am. And I think, it took me 6 months to make this library. And now, a student in my lab could do that in overnight, a couple of days. Yeah. And I spent my entire life, making packaging extracts and all kinds of stuff to make these libraries. And so it was very exciting because I had to develop my own way of doing things and how to differentiate the cells and how to screen all these phage libraries. And I cloned a couple of interesting genes, did a lot of manual sequencing, which of course doesn't exist anymore. So it was hard work, but I really enjoyed it. It was great.
So you were already interested in cell fate.
Read interest in self-fate and particularly in gene expression. I've always been interested in gene expression and how it's controlled and how it's regulated with sort of self-fate determination, how that affects gene expression. So that interest goes way back to the very beginning.
Then you did, after the PhD, you did 3 postdocs between 1985 and 1992. Can you tell us more about these years between 85 and 92?
Okay, yes. So when I finished my PhD, I wanted to stay in London, and there was one of the 2 labs I wanted to work in was Keith Willison at the Institute of Cancer Research, Chester Beatty Labs. And I got a postdoc with Keith, and I was working on the T complex. And I just got started, and my husband got offered a great job in Ninewells Hospital in Dundee in Scotland.
I see.
And so I had to just give that up and immediately go back up north. And so that was a shame because it was a great lab and I enjoyed it. It was a very short time. But then when I got to Scotland, I was really lucky because I got a postdoc with Ron Hay at St Andrews University. And Ron is a fantastic scientist. He's a real expert on NF-kappa B signaling, simulation, DNA replication. And so I learned a lot from Ron about, again, more about gene expression and transcription. I learned all that there at St Andrews. And then when I finished at St Andrews, and I had my first child there, I had a daughter while I was a postdoc, so that was a bit of a disruption, slightly, to my life. And then I moved to work with John Clark, the late John Clark, at the Rosalind Institute in Edinburgh. And that's really where it all began, with mammary gland.
So then is it there where you started to be interested in mammary gland biology?
Yes, because John was working on making lots of transgenics that would direct gene expression to the mammary gland, because his idea was to use the mammary gland as a bioreactor. So he had a real interest in making therapeutic drugs in the mammary gland. And he used the beta-lactoglobulin promoter, which is not expressed in the mouse, to drive transgene expression in the mammary gland. And so when I joined John's lab, my task really was to try and work out the essential promoter regions of BLG and how expression was regulated.
So these mice, the beta-lactoglobulin crates, were generated back then?
Way back then. Long time ago. So that's and we've used these mice now, ever since. So
Absolutely. There are several groups who are using them.
I think they're all around the world and they're very useful. I think they're very useful. So yes, so that's I then in John's lab started to look at transcription factors that would bind to the promoter of BLG, and that's where we discovered what we called milk protein binding factor, which is the same as Bernd Groner called mammary gland factor, which of course turned out to be STAT5, and that's how we got into stat signaling, JAK-STAT signaling, way back then.
So in 1992. You started as group leader in the Rosalind Institute.
Yes.
But before talking about your scientific career, can you tell us a bit about your personal history, your birthplace and childhood?
Okay, I'm Scottish, as I'm sure everyone listening to this can tell. I'm trying to moderate the Scottish accent just a touch. But I was born in Greenock, which is on the west coast of Scotland, on the River Clyde. And it's about 20 miles from Glasgow, so I was near a big city. And Greenock was a shipbuilding town. So one thing I remember from my childhood is all these fantastic ships being launched. And if you go to a ship launch, it's amazing. You hear these huge vessels gradually sliding down this runway and then hitting the water, and it's the most amazing thing. And so it was a great place to grow up. It was very much an industrial city. It was a place where there was a lot of work really to do with the shipyards, engine works, all that kind of thing. A lot of my family had a history of working in the shipyards. But it's the most beautiful part of Scotland, but the weather is so terrible. If it wasn't for the horrible weather, it would be bliss, because there's mountains, there's water everywhere. It's absolutely beautiful, but the weather is just terrible, I'm afraid. But my childhood was very ordinary, uneventful. I have a younger sister, so we grew up there together. And I liked school. I liked playing hockey. I liked playing tennis. I'm still in touch with friends that I made all these years ago when I was in Greenock. Still keep in touch. So it was an uneventful life, but very secure and happy, I think, as an early life.
So then, what made you go into science and not into art, music, hockey, horse riding, tennis?
I think, when I was little and at school, I had no idea what I wanted to do. All I knew was I was terrible at art. Anything but art, because I just couldn't do it. And it's funny, my sister is actually an artist, so she's fantastic at it. But then, when I got to secondary school, about the age of 11, I then discovered science. And I remember one of the first experiments, we had to take different liquids and put them in a glass measuring cylinder. So we had like coloured ethanol, mercury, would you believe? The 11-year-olds playing with mercury. And other liquids, oils and things. I would put them in the cylinder, and then of course they all separated out into different layers. And I was just fascinated by that. I thought, wow, amazing. And then I sat my first exam. I beat all the boys, and I thought, I just love this. I'm going to be a scientist. And that was it. I just knew right from then I had to do science. I mean, I started off wanting to be a chemist.
Okay. It's good that they showed you this experiment first, instead of asking you to do some drawing from the microscope.
Absolutely, yes. that came a lot later in my undergraduate years, having to do the drawings. Which was still pretty terrible,
yes.
I have to say. But no, I just loved I loved the chemistry, and I even liked the physics. Okay. I quite enjoyed doing all the physics as well. So that was it. I knew that's what I wanted to do. Never thought about anything else after that.
So as group leader, first you were at the Roslin Institute, then you moved to the University of Edinburgh, and in 1998 you joined the University of Cambridge, where you are today a professor of cell and cancer biology. Can you tell us about your first years as group leader?
Okay, so I became a group leader, as you say, in 1992, and this all started because the work I'd done with John looking at the BLG promoter, because we'd identified staph factors and other factors that regulated transcription, it was thought good enough to give me a personal 5-year fellowship to allow me to develop my career. So that was my career development fellowship. And I was awarded that 2 years after my son was born. So that was quite a challenge. I had to get back from maternity leave into the lab and set up my own group. So that was really quite a big challenge. But in a way I was lucky, because I was still at the Roslin Institute and I was still under John Clark's direction. He was head of the department. So he was very supportive, and that allowed me really to get the lab up and running. And I was incredibly lucky because my first postdoc was Tom Burden. And I don't know if you know Tom, but Tom worked with Lothar Henninghausen.
That's interesting.
And of course Lothar is such a fantastic mammary gland biologist, so Tom was really well trained. And it was so lucky that he came and chose to come and work with me. And that, together we were really a good little team to get going on BLG. And I also had a great PhD student, my first student, Katrina Gordon, who made the Kim2 cell line, which really means Katrina's immortalised cells. Katrina's immortalised mammary cells. And she developed this wonderful cell line that will differentiate, it will make milk, it will die when you take the hormones away. It's great. So we had a really good little team to get started on. And I think that was the very start of it. And then it went on from strength to strength. I think more people joined the lab, I got more funding, and things went really well, and I really enjoyed that. And then I was very lucky then that I then moved to work with Andrew Wiley at Edinburgh University. So we basically moved across town, and I went to work with him. And that's partly because by that point I was interested in cell death. So that was a really good place to go to work with him, because as I'm sure everyone knows, Andrew Wiley discovered apoptosis. He was one of the first to describe it. So,
how did they call it at the beginning? Do you remember? Was it called apoptosis from the beginning? Or?
I think they coined that term very early on. And I think because of this notion that it's a Greek word for leaves sort of falling, and that was, they wanted a separate word for it because it was a different type of cell death. They knew it wasn't necrosis, but it was a programmed type of cell death, and they wanted a specific name. So I think programmed cell death was around as a concept, but nobody had coined a term for it before. So they were the first to describe this DNA laddering type of phenotype, and they called it apoptosis. So the early years were great as a group leader because I was in a fantastic environment, surrounded by mammary gland biologists. And
students and postdocs.
Good students and postdocs. Edinburgh attracted a lot of very good people. Good facilities at Roslin.
Yeah.
And of course it's where they made Dolly the sheep, so there's a lot of expertise in transgenics and all that kind of work.
Sure.
So it was quite an exciting place to be as well.
Yeah.
So I enjoyed that very much.
And Dolly was made from...
Yes, from a mammary gland, absolutely. So the whole connection is there.
Exactly.
Yes, that's right. So she was made from a sheep udder
Sure.
cell. So yes, the mammary gland connection goes through.
And then you moved to Cambridge?
Then I was with Andrew Wiley in Edinburgh for a year, and during that time he was headhunted to Cambridge to be head of department here, and he asked me to move my lab down with him.
So then you moved all your lab, everyone?
So I moved, most of my lab came down here with me. I left Richard Clarkson behind for a year to run things in Edinburgh, tidy everything up, and he just had his first child, so it wasn't a good time to move. So he ran the lab in Edinburgh, and then he moved down here as well, and the lab was complete. So that was quite a smooth transition. It was quite a challenge, I think, trying to move a whole lab at one time.
So, Christine, who were your role models, if any, and who were your mentors?
Well, I don't think I have a role model, because when I was young, there weren't very many women in science. There's still not enough, I have to say, women at the top. But really, there was no one I could look up to and say, I would like to be like her, because there just weren't any women really around. So that was unfortunate, and I think today's young women scientists have a lot of role models, and I think that's fantastic.
But still not enough women, as you said.
But still not enough women, I think. But I've had good mentors, so I think my PhD supervisor, Peter Rigby, has been a great mentor. He's always been there to give me good advice. He tells me how it is, which is great, and he knows what goes on, and he's been a fantastic mentor. And also, he really knows how to run a lab, and I learned a lot from him in that sense. And so it's good to have I think it's good to have a mentor that goes way back, who knows what you were like as a young investigator, and who can follow your career and give you good advice as you're developing your scientific career. So I think that's really important.
So you remained in touch?
To remain in touch, I think if you can with your graduate advisor, I think is a really good thing to do. And also I think Andrew Wiley was a great mentor, because he really supported my work on cell death. He was a great enthusiast for that, he really supported that. And I have to mention Mina Bissell, because she's a wonderful woman, and I had a very short spell in Mina's lab, a sort of short sabbatical. And since then, she's been so supportive.
So you were already group leader when you...
I was already group leader when I went there. I had a few months in her lab, really to learn how to do sort of 3D cell culture. And I just thought she was amazing and very inspirational. And she's been good. I can email Mina if I want advice, and she will be happy to reply. And I think it's very good to have a range of people that you can mentor you throughout your career. I think it's very important.
So, Christine, your first paper on mammary gland biology was on interaction of DNA binding proteins with the milk protein gene promoter in vitro, identification of mammary gland specific factor. So this was in 1991. What did we know about this topic back then?
That's a long time ago, isn't it? Actually, we knew very little about the regulation of milk protein gene expression. I think Jeff Rosen and Lothar Henninghausen were interested in WAP and how the expression of WAP was regulated. Bernd Groner was very interested in beta casein, and we were interested in BLG, mainly because, of course, that was the emphasis in John's lab, and we had access to the promoter and to lots of transgenics with different promoter deletions. And I think really we didn't know very much more at that time, except, of course, there had to be some hormonal regulation, and there had to be some specificity for the mammary gland, but we knew very little at that time.
And so what kind of model systems did you use back then? This is before the mice, right?
We actually were able to define the promoter quite well with mice, and then we deleted binding sites for STAT5 in the BLG promoter and made transgenic mice. So this was partly work done with Tom Burden. So we actually made so many lines of mice and looked at BLG expression when we mutated the promoter. So that was a lot of work, very complex work. And I think other labs were doing similar things. But we also did in vitro work. So we could take promoter regions and we did electrophoretic mobility shift assays where we could bind the different nuclear extracts with different proteins to radioactive binding sites. And that allowed us to really define all the different members of the STAT family and the sequences they bound to and how important these sequences were for regulating gene expression. So yes, the techniques back then, now we have, with RNA-seq, it's somehow not easier, but much more comprehensive and much faster. It was a laborious process. I mapped a 400 base pair promoter in 50 base pair overlapping oligonucleotides. Did band shifts on every one of those oligos and mapped the whole promoter. And that took quite a bit of time.
Absolutely.
And then of course you can compete off with different binding sites to try and define which transcription factors are binding. Which I loved. I loved doing that. I've done more band shifts than anybody and I'm still alive.
I guess this is the influence of the NF-kappa-B work, probably.
So yes, the NF-kappa-B work, absolutely, yes.
You learned all these techniques, when you were working on NF-kappa-B, then you...
That's right,
wow.
That's right. And stats are beautiful to band shift. NF-kappa-B is a horrible thing to band shift, but stats are lovely, give you beautiful, beautiful shifts. So so that really, there wasn't a great deal known then at all. That's amazing what we know now.
So Christine, your current research is focused on understanding two essential processes for mammary gland development: commitment of stem cells to the luminal and basal lineages, and execution of cell death and tissue remodeling during post-lactational regression, what we call involution.
Yes.
So before we talk about stem cells, can we talk a bit about involution? So when did you start working on involution, and what did we know about this process when you started working on it?
So I started working on involution about 1995, and that really followed on from work, really, because we got so interested in the STAT family of transcription factors. By that point, we knew there were 7 different STATs, and then we undertook a study to look at how they were induced during a whole mammary gland developmental cycle. So we looked in virgin tissue, we looked at different time points in pregnancy, lactation, involution, and then we looked to see which STATs were active at different times during this whole process. And it still amazes me that you can clearly define these different stages by looking at which STATs are active. And they're very discreetly and distinctively switched on and off at different times. And so around this time also I was collaborating with Bill Miller in Edinburgh, looking at breast cancer. And we had breast...
He was in Canada, right? Bill Miller?
Bill Miller, not Bill Muller. A different Bill Miller. So the Bill Miller from Edinburgh, who worked a lot on sort of steroid hormones in breast cancer. So he's really a breast cancer biologist. And Bill had lots of biopsy material, and we discovered then that STAT3 was constitutively active in breast cancer extracts. So we were actually the first to show that tumours have constitutively active STAT. So that was way back in 1995. And because we discovered then that STAT3 was active at very high levels during involution, that then prompted us really to start looking at involution. We thought there's a transcription factor here, it's hugely upregulated, very specific at this time. There must be a lot of gene expression changes going on at this time. So that's why we became very interested in sort of STAT3 and what it was doing. Up until then, people knew caspases were activated, they knew that there's a lot of beautiful old electron microscopy work done looking at mammary gland involution, which people forget about these days. But actually, it's amazing. If you go way back in the literature and look at the electron microscopy work, there's a lot of stuff out there that was really kind of known and seen already, but the modern tools to look at it were not available.
Yeah, and the molecules that were involved were not known.
Absolutely, but people had a really good idea about
process.
Some of the processes in terms of how that looked for an involuting tissue. So really, I think most of the work was very much based on the original electron microscopy, and then we came in, we were interested in STATs, and we knew then that cells died very rapidly following forced involution. And it's an amazing rate of cell death to see these cells dying, and they had an unusual morphology, which was also sort of very interesting to us at the time. The other things I guess we knew that the local factors were important, because Priscilla Firth had done some very nice work with teat sealing, and shown that it's not the circulating hormones that affect involution, but it's local factors. Because if you seal the teats on one side, those glands will undergo involution, but the ones with the unsealed teats don't. And they're still being exposed, everything's still being exposed to prolactin and all the other hormones. So we knew it was a local factor from her work. And we also knew that there were two phases from work that Zina Werb's lab had done, showing that you need proteinase activity really to completely remodel the gland later on after about day 3. But early on you don't need this activity. So she suggested there's two quite distinct phases of involution.
So
we knew these things.
Yeah, that's fascinating. So what do we know today?
Today, oh my goodness, what do we know? I wish I could say to you we know the answer. That, you know, we could absolutely tell you everything. And we're not there yet. And I'm hoping before I retire, which is a little while away, I will be able to say to you, we have answered it, we know exactly. Because I think we can do that. I think we can get there. At the moment, we know lots of things are important. You can knock out lots of different genes and they will all affect involution. But I think for me, the overriding factor is definitely STAT3. Because STAT3 seems to coordinate a lot of different processes. We also know that NF-kappa B is important, because it's probably the other arm, and you need both arms to really be activated to kill a cell properly. But if you take out STAT3, which we've done with conditional gene deletion, you can delay involution, you can take out the whole first phase of cell death, and in fact Lothar showed that you can delay that first phase for up to 6 days. So instead of being a 2-day delay, you can now have 6 days. And if you give the pups back to the mother, the gland will make milk again, which is remarkable. So I think STAT3 is probably the most important.
So in the normal situation, in the white-eyed mouse, if we put back the pups before 48 hours, the mammary gland will resume and...
Absolutely, and it makes sense, doesn't it? Because in the wild, there's a good chance that the mother might be away from the nest for a certain period of time. And not get back, and of course it would be disastrous if the gland underwent involution irreversibly. And it's interesting, in other species it's a different length of time. So it's much longer in cows, which is estimated to be like 11 days. And Kevin Nicholas in Melbourne has done nice work on fur seals, which swim away and leave the babies behind for about 3 weeks at a time. And they can come back and still lactate. So they can keep that reversible phase for 3 weeks. Which is remarkable.
And in human, is it known?
I don't think it's known. It's difficult because humans tend to involute gradually because they tend not to force wean, although it does happen. But I'm not aware of any studies where women have tried to wean at maximum lactation and then try and recommence that. But I guess it would be maybe several days, I would have thought, probably. But I don't know. The longest I was ever away from one of my children was 12 hours, so... It's definitely longer than that. I don't know how long it could be, but anyway.
So besides STAT3, what are the factors, the main players? Or you would say that STAT3 is the major one? Or?
I think we've shown that STAT3 regulates so many things. So not only does STAT3 regulate the whole cell death pathway, because now we know that STAT3 regulates a lysosomal pathway of cell death.
Absolutely. And
it upregulates cathepsins, it probably upregulates the lysosomal compartment, it also downregulates the expression of a gene that is an inhibitor of cathepsins. So it's upregulating the killing pathway, downregulating the negative regulator of that. It's also switching mammary cells from being secretory epithelial cells to start behaving like phagocytes. So it's been known for quite a long time, apart from Peter Henson and other people in Denver, that mammary epithelial cells can actually eat up the dead cells and the milk that's been shed into the lure. But we've shown that if you take out STAT3, these cells do not undergo this switching to be like these particular M2 type macrophages. And they don't express a lot of the markers like CD14 that would say these are phagocytes. So STAT3 seems to coordinately control not just the death process, but actually probably the switching of the epithelial cells to become more phagocytic in behaviour. And so, I mean, if you knock out STAT3, there are hundreds of genes that are now no longer upregulated. So it's a very complex process. STAT3 also regulates the acute phase response, inflammatory signalling. It seems to coordinate the whole sort of second phase of involution by upregulating things at a much later kinetics, slower kinetics. So I'm a big fan of STAT3.
Yeah, so what happens to NF-kappa B? I remember work from your lab a few years ago.
Yes, well, we showed that if you delete I-kappa B kinase beta, which is the upstream regulator of NF-kappa B, you can also delay involution. And that was a surprise to us. We thought NF-kappa B should be a survival factor.
Sure.
But this kind of suggested it wasn't, that it was a killing factor. So actually at the moment we're now knocking out both simultaneously to see if that really takes out cell death completely. I can't tell you the answer to that just yet. But we'll know by the summer, I think. So we haven't done a lot more work on the NF-kappa B arm, but we know that's also a regulator of inflammatory signalling. So often STAT3 and NF-kappa B go together. They both regulate inflammatory signalling in lots of different tissues. What that does in terms of involution, I'm not too sure, but might be really important for the whole remodelling process, which is like an inflammatory response that's kept under control.
Yeah,
or
so
like a wound healing response.
Yeah, so this is bringing us to the next question I wanted to ask you. So, what are the links between involution and cancer?
Indeed, and that's such an important question in a way that we don't know very much about it. I think, as I've mentioned, we know that STAT3 is active constitutively in cancer, in many cancers, and also in many different types of breast cancer. And that's a bit of a paradox. So why should a killer in the normal breast be actually a survival factor in breast cancer? And many breast cancer cells are addicted to STAT3. If you inhibit STAT3 activity, they will just curl up and die. And it's a big, big paradox. And I think in a way it's easy to resolve that paradox because you can say that breast cancer cells are not differentiated the way lactating cells are. They're not terminally differentiated. They're not really secretory epithelial cells. And they're going to respond differently to the STAT3 signal. They don't have the other components, perhaps, of the machinery that they need to undergo cell death. But also, we know from some of our work that a lot of the STAT3 target genes are not induced in breast cancer. So some of the genes that STAT3 would regulate in mammary glands are absolutely not upregulated by STAT3 in breast cancer. And these genes might be quite important. So there is that connection, I think, with STAT3. And the fact that STAT3 induces this inflammatory response, and we all know now the tumour microenvironment is so important for cancer. We've done xenograft experiments in mice and shown that cells that don't normally express or activate STAT3 will activate it once they're in the mouse making a tumour. Yes. So they're signalling from the microenvironment to these tumour cells, and they now activate STAT3. Yes. And of course we then get reciprocal signalling from the tumour cells back to the microenvironment. And that will bring in macrophages, all kinds of inflammatory cells, and start then remodelling that environment and promote sort of tumour invasion. So I think there's a real connection there. Yeah, the other connection I think is more direct, and it was really pointed out by really elegant work done by Pepper Shadan's lab, where she showed that if you take matrix, extracellular matrix from involuting mammary gland, that can promote tumorigenesis. And then she then went on to demonstrate that involution itself can promote in xenografts more growth of breast tumours. So there's a more effective growth of the tumours if the mice have gone through an involution. So that's really interesting. And Pepper's very interested in pregnancy-associated breast cancer. Yes. And it's this subtype of breast cancer that women get after pregnancy or a period of lactation, up to about sort of 2 to 5 years after that, they're more susceptible to breast cancer. Yes. And there's a very good suggestion that might be because of this inflammatory microenvironment that cells are exposed to during involution. So you can imagine if you've got a very tiny lesion in the breast, you then undergo an involution, these cells are now exposed to this massive tissue remodelling inflammatory environment. Sure, yes. And that perhaps could promote changes in those cells. So we're also quite interested in that, and that's something we're looking at more from a sort of perspective of the inflammatory microenvironment in involution. And so we have a project looking at that as well. So I think that's going to be very interesting to see how that pans out.
So I know you are also interested in stem cells. So how did you get into mammary stem cells, and what fascinates you in this area?
So I think I've always been interested in stem cells from my PhD days, because I was interested in the embryonal carcinoma cells. So I've always loved this notion that you can take, one cell, which is a fertilized egg, and that can then, and I tell my students when I do lectures on this, that you have one cell and it will make every cell in the body, and that's all down to the control of gene expression, basically. And so I've always been fascinated by how you can take a cell and then differentiate that cell into lots of different types and different lineages. And quite a few years ago now, I think probably back in 2007, I was joined in my lab by Lisa Anderson, who came to do a PhD with myself and jointly with Gil Smith at the NIH. So this was a programme to share students between the NIH and Cambridge. And she was funded on that programme, which is great. Sure. So it means that you can have a close collaboration with somebody at the NIH. Yes. And of course, Gil and I had known each other a long time, and this seemed like a perfect thing to do. And of course, as you know, Gil was really into stem cells. So when Lisa came to the lab, she was a very bright student, and she knew how to do fat pad transplants, because she'd done lots with Gil, and was very skillful. So that got us into doing transplants. It opened up techniques to us that we hadn't really done before, then opened up whole new areas for us to start looking at stem cells and mammary glands. So Lisa did some very nice work when she was with Gil and I doing that. And then John Stingle joined my lab for a couple of years, and John, of course, was one of the first people to use FACS sorting to isolate mammary stem cells and do transplantation of those cells. And so we had a lot of fun with John in the lab, learning how to FACS sort. Yeah. Also trying to squeeze him into the lift to get up to our lab because he was far too big. He's very tall. Those of you who don't know John, he's very tall. But we had a lot of fun doing that. And so that kind of got us into doing the FACS sorting as well. And coupled with our new interest in
this was after transplants. The publication of his Nature paper.
That was after the publication of his Nature paper.
2008 or 9. Paper was in 2006.
And
then he came a year or two
I guess
after.
it might have been 2008. Okay. He wanted to come to Cambridge.
Oh, lovely.
And we wrote a grant together, which was funded by Breast Cancer Campaign. So we're very grateful to them for the funding. And then thereafter, of course, John is still in Cambridge. He's just up the road from us now. So we can still collaborate with John very easily. It's a short bike ride away.
Absolutely.
So that kind of got us into doing slightly different things as well. And people in my lab now who are still interested in doing stem cell work. So we're kind of split. Kind of 50-50, bit more on the cell death, which I guess is my first love, but also we have people in the lab now doing stem cells and self-fate, and we have a stem cell institute literally just over the road there, and we're affiliated with the Stem Cell Institute, and so we have rotation students come through the lab who are interested in stem cells. We interact a lot with people there, so the head of the institute is Austin Smith, who's of course well known for his groundbreaking work on embryonic stem cells, and so we have very good people to interact with in the stem cell area. And there's quite a lot of epithelial biologists there as well, working on skin and other tissues. So.
Well, and there is mathematical modelling by Ben Simon.
Absolutely right, that's right, and there's a lot of people working on there, people like Doug Winton up at the Cambridge Research Institute working on intestinal stem cells, so we interact with Doug Winton, and lots of people in Cambridge. To do stem cells here is not, it'd be crazy not to do stem cell work here. There's so many opportunities.
Absolutely.
And there will be a whole big new stem cell institute being built up at the hospital site in a few years' time. So it's going to be a great place to do stem cell biology. So so we're very excited. We go across to the stem cell meetings and all the various things, and so we're going to keep up that interest. But I think for me, it's not so much about perhaps the stem cells that set up the gland in the first place. Not so much the embryonic stem cells that give rise to the ductal structure. I'm much more interested in the cells that really give the ovular lineage, because that's a lineage that will have to keep renewing throughout adulthood. It's a lineage I think we don't know really a great deal about.
Absolutely.
And I'd love to know, is there an ovular stem cell? We don't know. So I'm kind of more into self-fate rather than directly stem cells, but we have a great interest in that too. So yes, exciting stuff.
Absolutely, and stay tuned, because maybe there will be studies coming on alveolar stem cells. At the junction of the TDLU, the terminal ductal lobular units, that's the area where many of the cancers arise.
Absolutely right, and that's where the alveolar progenitors are probably sitting.
So it's plausible that these cells are sitting there, and these cells could be the target or one of the targets of neoplastic transformation.
I think you're absolutely right. You know, one thing we're looking at now is looking at the cell of origin in different cancers and looking at ablation of particular lineages, so we can see which lineage contributes to which particular type of cancer. It may be that this won't work, it may be the mammary gland is so incredibly plastic.
Plastic, yeah.
And I think it is really plastic. So this kind of study might not be terribly revealing. But we have to look and see.
So stay tuned.
Yes, absolutely. And in 5 years' time, I think the whole field will have changed again. I think this field changes so quickly.
Absolutely.
It's going to change again. There's going to be the next big leap forward very soon, I hope.
So Christine, you mentored many students and postdocs. So how important was this for you?
You know, in some ways, it's almost the most important thing I do. I love the research. I get so excited about the science and the data. And I still, I mean, before you came, I was looking down the microscope because I wanted to see the data for myself. And I still love that. But in many ways, one of the wonderful things is to have a young person come into the lab. Somebody who perhaps is lacking confidence or research skills, but who's got an interest in science. And to take this person and to foster them. And at the end of 4 years, to turn out into the scientific world somebody who's now a fully fledged scientist, who has all these research skills, who has the confidence, the ambition, you know, the background now to go off and start doing their own research. And I find that incredibly rewarding. I've had some wonderful students in my lab. And in fact, 2 of them recently have got faculty positions in Cambridge.
Oh, lovely.
So that's made me very happy. I think that's the ultimate for me, to have my students go on to actually be faculty here at Cambridge as well. And it is one of the things I love most of all, is to see, it keeps me young as well. You know, you have all these wonderful, bright young people come into the lab, full of enthusiasm. And that for me is what science is all about. It's not about the big successes out there, the big group leaders. You know, it's great to talk to them, but really, you know, the young people who just have... Absolutely, you know, they just have so much enthusiasm.
Yes, absolutely.
And I love that. And I think one of the things I learned from my PhD supervisor was that science has to be fun as well.
Absolutely.
We partied a lot. So Peter would give us money to go and buy all kinds of things to party with. And I think my lab's a fun place to be. And I think that's really important, that people learn that science is fun.
Absolutely.
It's a serious business. You have to work hard, but it has to be a lot of fun. So I hope I instill in people this wonderful enthusiasm for research and the skills to do it, but I also hope that they've understood that it has to be fun, you have to enjoy it, and you have to really think about all the people around you.
I think great scientists take their science seriously, but they don't take themselves seriously.
I think that's a wonderful way of putting it. I absolutely agree with you. I think that is definitely, I think, the right attitude to have to science.
So do you teach?
I teach, yes I do. I have quite a lot of teaching this year. In fact, I have a lot of teaching next term. So I teach cancer biology, of course, to the final year students here. And I'm also, for the first time this year, teaching the first year medical students about cell cycle, which I don't know enough about, and cell death, which I'm a little bit more in my comfort zone there. And that's quite intimidating, because there'll be about 300 of the brightest young people in the UK are going to be listening to my lectures, and so actually I find that more intimidating than talking at a conference. Because you know, at a conference you know what you're talking about, and you have reasonable expertise. But you know, to talk to students like that who somehow are so bright that they will come up at the end and ask you the really challenging questions and test your knowledge to the limit.
And today they can take their phone and then check and ask questions.
Absolutely, yes, that's right.
But did you see the last paper? So,
indeed.
The French philosopher Montaigne said, teaching is not about filling a vase, it's about kindling a fire.
Indeed, and I like to say to my students, I'm not here to tell you anything, I'm here to inspire you. Because that's what I want them to do. I think students here do not need to be taught everything. I think they need to be given the idea of what the subject is all about, and they need to be inspired to go and read about it themselves and really make it their own knowledge. So easy just to regurgitate what someone has told you. But if you find out for yourself with guidance, then you really understand it, you really learn it.
And you remember it better.
Of course you remember it better. And one of the things that we have in Cambridge, which is fantastic, is the supervision system. So all students in Cambridge are in a college, and the college will provide extra teaching for students on this subject. And what I do for the final year students, because they don't get this extra teaching, is if they ask me, can I give them a supervision, then I would say yes. And we get a group of them together, and we can sit down around a table like this, go through my lecture, they can ask any questions they like, and we can have a really good conversation about the topic. And I think that's a really valuable addition to the teaching we have here.
Absolutely.
Is that they can talk to somebody who's actually doing the work and can answer questions directly. And I think it's great. I think it's really important that students are inspired to do that kind of thing.
Cambridge has a long tradition of transmission, I think.
Yes, I think that's right.
So, Christine, what would you advise a junior faculty who is going to start her or his team to work on mammary gland development or cancer and cancer?
Well, first of all, I would say, yes, do it, because it's a wonderful field. I love it. I'm so glad I got into mammary gland biology. And yeah, I mean, I think it's an important field though, because mammary gland is wonderful. You can understand about cell death, you can understand about stem cells, you can understand about differentiation, hormones, anything. The mammary gland is a wonderful model. But of course, breast cancer is such a devastating disease. It affects so many women and men. Men get breast cancer too. And it's thought that we will have an epidemic by 2050 of breast cancer. So although we can detect cancer earlier, we still can't really cure it. Women survive a very long time with many of the treatments we have, but a lot of them still become resistant. And there's a lot of work still to be done. So I think it's a great field to get in. There's quite a lot of funding because breast cancer is such an emotive subject. There is funding. There's a lot of charity funding. Particularly in the UK, there are breast cancer specific charities. I know there are in the US as well.
Sure, yes.
There's a lot of charities specifically for breast cancer.
cancer.
So there is funding. I think there's less funding for the basic biology. Yes. Is it a shame? I'm a big believer, as we talked earlier about blue skies research. I think you can't translate something that you haven't understood in the first place. So I think we need to have more funding for basic research. But I think stem cells, again, are a very interesting and emotive area. And I think there's a lot of funding for stem cell work. So I would say definitely get into the field, but when you do that, I would recommend going to the mammary gland Gordon conferences.
Oh, absolutely.
Get to know everyone in the field, and what is great is that the top people still come to these meetings, get to know them, establish collaborations, and really get to know people. And don't do it on your own, really get to know people who are expert and who will advise you. Because it's a field where people will help you.
Absolutely, and at this meeting, it's not kind of cliquey meetings. There are no cliques. You just sit there for lunch or breakfast, and then you turn. Oh, this is the. I remember I was my first year of postdoc, and then I was sitting next to a gentleman, and he turned. Oh, I'm Jeff Rosen. Nice to meet you. Who are you? What do you do?
Yes.
And then you end up talking with the greatest scientists.
Absolutely, and I would recommend that, because you can go to other meetings where really a lot of the speakers don't associate.
Or don't stay.
Or don't even stay, that's right. So I think it's quite a complex tissue, the mammary gland. I think you need to have knowledge, and I think for that it's good to interact with people.
And to know also the history.
And indeed, absolutely.
And I think this is one of the goals of these conversations in mammary gland biology and breast cancer.
To know where it's all come from and how we all got started. Absolutely.
So Christine, what are the major questions that you think still need to be answered in the field of mammary gland development?
The big questions. Well, I think we'll probably need to talk a little bit more about this, but I think we don't know about mammary stem cells. We really don't. We don't know where they are. We can't identify them. We have no markers. We really don't know anything about them. We don't know about lineage commitment, really. We're way behind fields like the skin and the hematopoietic system, the intestine, where people know where the stem cells are. They know how quickly they cycle, they know where they go. There's a lot of that is known, and we just don't have that in mammary gland. And I think there's still a lot we really don't understand. Why is an early pregnancy protective against breast cancer? Absolutely. We don't know that. It's such an important question, and we don't know. And I think we really need better models. We still don't have good models. I think, of breast cancer. I don't think we have good cell culture models, really. I think we need to develop these better. We need
more
three-dimensional models.
Cultures.
Heterotypic cultures, absolutely. It's one thing my lab is doing at the moment is we're developing engineered scaffold structures where we're actually putting in epithelial cells, macrophages and adipocytes in co-culture to try and look at tissue interactions.
Yes.
But it's going to be very hard to make good model systems, but we will need that. So I think there's a long way still to go, I think, which is great because it's going to keep us really busy and fill us with excitement, but I think there's still such a lot we don't know.
So what are the important questions regarding involution?
Involution. I think involution, one thing that is really interesting is why some cells die and some cells don't. The ducts remain.
Sure.
Why do the ducts not die? We don't know. We really don't know. I have some ideas, but we don't actually know why that is. And it's, I think, very interesting how the gland can go through this sequential process of death as well. So what is this switch to irreversibility? We've tried looking at that. I had a fantastic student who tried to look at this and we just couldn't get the answer. It's actually very difficult to do these experiments, but we don't know what that switch is that says, okay, we can't stop the death now. That's it. Everything's on the road to destruction. We've got a lot of markers of that switch point, but we don't know what is driving it. And I think that's very interesting as well.
Could it be that when the neighbor cells or when stromal cells think that the epithelial cells are dead, and then they
it could be that it gets to that, the numbers of cells are too low, or perhaps the dead cells have sent out signals to the macrophages, and which are now coming in and
the balance of macrophages.
Macrophages are now killing, basically. It could be, or not, actually triggering breakdown of the extracellular matrix or something that's triggering more of the death. So that would be very interesting. But I really would love to be able to define involution and say, this triggers involution.
Absolutely.
This is the most upstream event, and this is exactly how it's all controlled.
And this is the point of non-return. This is the mechanism that causes the point. Yeah.
And why the ducts survive, and why alveolar progenitors or stem cells survive.
Yes. Because
in the mouse, certainly we know that there can be 20, 30 cycles of development during pregnancy.
Sure.
It's inexhaustible. So what are these cells that give rise to the whole alveolar structure the next time? They have to be protected somehow from dying.
Well, these are very important questions for in the stem cell field.
Yes, that's right. That's right.
So thank you very much. I have still one or two more questions.
Okay.
What would you like to tell the next generation of scientists who will watch this video?
The next generation of scientists. I think I'd like to say, it is the most rewarding career you could have. It's not easy. I think it's very challenging in many different ways. It's difficult to keep the same career going over, I've been doing this now for 35 years or something, quite a long time. But I would recommend it as a career. You have to have tenacity. You have to be prepared to take the knockbacks when things don't go well or you don't get funding. But you have to be determined and see the way ahead and be prepared to work hard and to interact a lot with other people, but also look forward to an exciting career. But it's never going to be easy. I think it's harder now. I feel really fortunate that I started when I did. I think now it's much more difficult for young people to get on that career ladder and to really establish themselves. But I think you have to look for your niche. You have to find an area that is yours and that no one else is actually working on, that gives you that potential to be a leader, to drive forward something quite different. So yes, I think if you want to be a career scientist, definitely go for it. It's a wonderful thing to do. But you've got to be tough, I think. Be prepared to take the hard times as well, definitely. But I've loved it. I've loved every minute of it, and I would never, if I could have my life again, I would still do it. I wouldn't change it for anything.
So Christine, is there any question that I should have asked, but I did not?
I think the only question you haven't asked is there one person I couldn't have done this without? And I would say to you, that has to be my husband Gordon. He may never watch this video, but I'll say it anyway. He's been absolutely wonderful. He's been supporting me now for 38 years. And it's a bit of a roller coaster ride having to live with a scientist. It's not an easy thing to do. And I think he's been fantastic and very supportive. So I think I owe a lot to him as well. And I think everyone needs somebody to support them through a career. I think the rocky road of science. I think everyone needs someone to really support them.
Great.
And he's been wonderful.
Thank you very much, Christine. It was a pleasure for discussing with you about your career and about our field. Thanks very much.
Thank you, Momo. It's been a real pleasure talking to you, and I really enjoyed it. Thank you for this opportunity. It's been great.
Thank you.
Thank you.